Good morning. So I'm going to give a brief update on the AZD 5847 development plan or project. So this is another oxazolidinone. These compounds bind to the 50S ribosomal subunit and inhibit the initiation of protein synthesis. This is not a new mechanism to antimicrobials, as linezolid has been approved for gram-positive infections for a number of years. And that compound has also been blazing the trail for this class in tuberculosis as well. There have been a number of studies recently, including a controlled phase 2 trial in South Korea that have shown very potent activity of linezolid against XDR-TB. However, the safety profile of that compound leaves much to be desired. And we'll discuss that in a little bit. But it's really the hope of this next generation of oxazolidinones that we can reproduce that efficacy, if not improve on it in some respects, but really, most importantly, improve on the safety profile moving forward. So the therapeutic concept for AZD 5847 really is wide open to the treatment of drug-sensitive or MDR, XDR, TB, and we also have the potential of treating HIV co-infected population since the CYP profile of the compound is quite clean. This program originally started as a development program for staph infections. However, the phase 1 results did not support QD IV dosing, and so that program was discontinued in 2002. However, a couple of years later, the compound was repositioned for the treatment of tuberculosis. And you can see that the distribution of MICs across a panel of 163 TB isolates is very consistent, and that covers a spectrum of drug-sensitive through XDR isolates. So in the next few slides, I'm going to show a little bit of preclinical data on AZD 5847, specifically around attributes of the compound that we think may help the compound differentiate in the clinic going forward and really stand out in the OXA class. And the first of those is activity against intracellular tuberculosis. So in a bone marrow-derived macrophage model, we see a very clear concentration-dependent decrease in colony-forming units over 10 days in that assay, whereas our benchmark within the OXA class, linezolid, does not show a significant decrease in that same assay. The next is activity against slowly dividing tuberculosis. So in this case, we're looking at a mouse lung infection model where treatment of infected mice is initiated either at 3 days or 28 days post-infection. And this is aimed at either early rapidly dividing bacteria or a more persistent slowly dividing population in an established infection. So if we look at the open symbols on the right, we can see that both AZD 5847 and linezolid show a strong correlation of increased efficacy with an increased exposure in the mice. However, the solid symbols show us that linezolid sort of tails off around 1 log, regardless of an increased exposure. However, AZD 5847 appears to remain in the same PK/PD index that we have for rapidly dividing bacteria. So this could indicate a better capacity for sterilizing activity. Finally, human mitochondrial protein synthesis. So MPS activity has been associated in the literature with a number of toxicities. This activity is present in all of the oxazolidinones. It's really just a matter of finding the most optimal safety window we can in dealing with this going forward. So this attribute has been associated with some of the most prominent adverse events that are seen with linezolid dosing, and in particular neuropathy. So if we look at this data for linezolid, and this is showing the original dosing regimen for gram-positives. I should note that recent studies have shown that in tuberculosis, there is the option of once daily or even lower doses of linezolid to treat tuberculosis. However, MPS does remain a real risk for the compound. If we look at equivalent data for AZD5847, we see that there's quite a lot of, all of the phase 1 doses are here, but there's a considerably lower exposure relative to the IC50 for MPS, including at 400 mgs. We remain below that for the entire dose. And furthermore, if we look at free concentrations, which is often the metric when assessing safety margins, AZD5847 is highly protein bound, and the free plasma exposures give us a multifold window even between Cmax at therapeutic doses and the EC50 for MPS. So we believe there is good potential for an improved safety window with linezolid, with AZD5847. So I won't go into a lot of detail here, but we used the phase 1 data along with preclinical PK/PD models to predict the therapeutic dose. One thing to note is that these models and all future dosing recommendations are based on dosing with food. So a crossover study that we did in phase 1 showed a dramatic food effect in exposure for AZD5847. And so based on that, we've predicted a therapeutic dose of AZD5847 of either 400 mgs twice daily or 800 mgs once a day. In phase 1, we were also able to show that the compound is safe and well tolerated through 14 days dosing at concentrations considerably higher than the predicted therapeutic dose. So the top dose was 1,200 mgs twice a day, or 2,400 mgs total for 14 days. There were no serious adverse events. The most common adverse event was dose-related nausea. However, that was primarily in the fasted group, so conveniently that's reduced by dosing with food. We saw white blood cell and reticulocyte observations in the highest doses only, as well as transient myalgias that were noted in a fraction, again, of only the highest dose that we administered. And there were no significant findings, ECG findings at all at any dose. So in the last, I guess, roughly the first half of this year, we've spent characterizing a carboxylic acid metabolite of AZD5847 that we saw in the phase 1 samples. So this was of interest since it's a relatively high exposure. The total AUC of the metabolite is approximately 50% that of parent. We did see this metabolite in the preclinical species that we had used for 28-day tox. However, it was at a considerably lower fraction relative to the parent. So technically, we did not have in vivo qualification. While we don't really need this until phase 3, we felt that we should at least get a basic in vitro safety package before moving ahead. So we synthesized and looked at the purified metabolite. We showed that it's equipotent against TB and has a similar kinetic profile. There's no structural alerts for gene tox. It's negative in an Ames assay. It does have MPS activity as well. However, it's less potent than the parent, so that's not really an issue. And we ran the metabolite against a secondary pharmacology panel of about 280 different enzymes, including a number of cardiac channels. We did see some differences in the profile across all of those enzymes. However, nothing that raised any real flags to make us feel that there was a change in the risk-benefit ratio. So we shared this data package with the FDA in a briefing document, and they were satisfied both with the data and with our plan to move ahead to phase 2A. So this is the phase 2A. This is a 14-day EBA study, which will be sponsored by NIAID. It will be performed in Cape Town by Andreas Diakon's lab. And I won't go into all the details of the study because it looks very much like other 14-day EBA studies that you've seen in the last couple of years. There are four different dosing arms and a RIFA4 control arm. And all of the details of this study are on clinicaltrials.gov. So next steps, we're really trying to focus on positioning the program for phase 2B now. And as such, we're gearing up to do 90-day chronic tox studies in mice and in dogs. So we've switched from rats to mice after looking at various species that would give us a better exposure of the metabolite. And we found that mouse, in fact, do give us a high enough exposure of the metabolite such that we will have in vivo qualification following this study. We will also be doing DDI studies in preparation for phase 2B, and the specific studies that we'll do will depend on the final design of that phase 2B study, which is still under discussion. And we'll be going into the phase 2B study using a capsule formulation, and this is really just about managing costs as we move forward, but we do recognize that to move to any studies, beyond that, we will need a proper tablet formulation. So we recognize that we have some pharmaceutical development work ahead of us as well. And so finally, in summary, we believe that oxazolidinones will have a prominent place in future novel combination regimens. We think that AZD5847 does have the potential to differentiate within this class when it gets to the clinic due to a number of preclinical attributes that I've discussed. It was safe and well tolerated in phase 1, and we're moving into phase 2A literally in the next 4 to 6 weeks. So hopefully a year from now I can share those results with you. And that's it.